Good morning, everyone. I'm Yunxiang Chen from the Pacific Northwest Nation Lab. I'm glad to share my work about how stream bed sediments affect the exchange flows and microbial respiration in hyperic zone. The interactions between sediment flow and microbes are very complex. To understand the interactions, we need to answer two questions. The first one is how can we measure the sediment structure, and the second one is how can we quantify the interactions. Here we propose to combine structural motion photogrammetry and stability modeling to answer the two questions. Here, this figure shows the workflow of structural motion, which generates millimeter to centimeter scale resolution riverbed topography. We then use this topography as the boundary between the surface water and the groundwater. We solve the surface water using the large eddy simulation, solve the groundwater using the dust flow, and then we solve the respiration using our reactive transfer model. Our iterative process is also applied at the riverbed in order to obtain continual boundary conditions for velocity, pressure, and concentrations. With this model, we can obtain the dynamic pressure, exchange fluxes, flow resistance, and the concentration at the riverbed. Due to the random nature of the dynamic pressure and the exchange flux, we quantify the fluctuation magnitude of dynamic pressure and exchange flux using their standard deviation. With this data, we further provide the linkage between the flow diameter and the sediment sizes, depth, velocity, and permeability, as shown in these three formulas here. We also evaluate how surface porosity affects the distributions of oxygen at the riverbed. If you are interested in this work, please see the full version of our slides. Thank you.